Several Numbers episodes were filmed at the Athenaeum, which is perhaps the most formal place on the Caltech campus where faculty and students and staff gather. My family has had several lovely celebrations there over the years as well. Hello, today we're going to talk about targets for recreational drugs. A reminder that nobody associated with this course condones the use of drugs that might be considered recreational in any way. Here's a diagram of a synapse and of the targets for recreational drugs. We begin here with amphetamine and cocaine, which act on neurotransmitter transporters. Amphetamine, in addition, may act on the neurotransmitter transporters in synaptic vesicles. We've discussed that. LSD, morphine, and heroin, and tetrahydrocannabinol all act on G-protein coupled receptors. Alcohol has many effects, which are mysterious and somewhat nonspecific. We're going to emphasize their action on G-protein gated channels. Caffeine is anomalous in one way. Most of these drugs are so effective because they're acting on molecules expressed on the surface of neurons. Caffeine does act on a GPCR, but its main and most general action is on an intracellular enzyme, as we've seen. Phencyclidine and nicotine act on ligand-activated channels. So let's do the details now. We're going to talk about the pharmacological classification of each of these drugs and the endogenous ligand that they might be displacing or mimicking. For instance, morphine and heroin are agonists, and they are replacing the action of the endogenous morphines, the so-called endorphins, which are peptides. THC is an agonist. It is acting on the same pathway as anandamide. Nicotine is an agonist. It's acting, as we know, on the same pathway as acetylcholine. Cocaine is an antagonist, acting in the dopamine pathway on molecules that bind and transport dopamine. Amphetamine and derivatives are both antagonists and false substrates, and they can act like noradrenaline or serotonin or dopamine. Ethanol is tough to categorize, but one way of describing it is that it's an agonist acting as though it were a G protein on certain channels. LSD is an agonist, and it's replacing the action of serotonin. Caffeine is an inhibitor, and it acts on the pathway involving cyclic AMP, again intracellular. Phencyclidine is an antagonist, and it acts on the pathway, a pathway involving glutamate. Now let's talk in detail about the primary target class and the details for each of these drugs. Morphine and heroin act on a GI-coupled receptor, a GPCR that activates GI, the inhibitory G protein. And specifically, that receptor is the mu-opioid receptor. Indeed, as I've told you before, mu stands for morphine. THC acts on a GPCR, also of the GI class, called appropriately enough the cannabinoid receptor. Nicotine acts on an agonist-activated channel. Several nicotinic acetylcholine receptors. Perhaps the one that's most important is the alpha-4 beta-2 nicotinic receptors. Cocaine acts on a plasma membrane neurotransmitter transporter, the dopamine transporter, which we've also called DET. Amphetamine and derivatives act on both vesicular and plasma membrane neurotransmitter transporters. We saw this in a previous lecture. The vesicular transporter that's most strongly affected is the vesicular monoamine transporter, also abbreviated VMAT. Ethanol has various actions. We're following its action here on a potassium channel, a G-protein gated inward rectifier potassium channel, which we've discussed previously in this course. It is actually a homomultimer of 2 GERK1 subunits and 2 GERK2 subunits. LSD acts on a GQ-coupled GPCR, the serotonin 5-HT2 receptor. Caffeine acts on an enzyme, cyclic AMP phosphodiesterase, which we've seen in a previous lecture on the G-protein pathway. Phencyclidine acts on a ligand-activated channel, the NMDA glutamate receptor. Now, how do we know all this? Well, there are various ways to discover the target for a drug. One way is the use of knockout mice. So let's say that you have the hypothesis that the response to a drug requires your favorite molecule. And so the researcher obtains the gene, the DNA, that encodes that molecule, and interrupts the gene with a detectable protein so that we can detect the interruption. And very often the detectable protein is the green fluorescent protein GFP. So that's why we've made this section green. So in some mice, then, the mouse gene has been replaced with the altered gene that encodes GFP, and those mice fluoresce green. And the researcher needs to select those mice that have been interrupted from those mice that have not had their gene interrupted and do not fluoresce green. When a good knockout founder mouse has been identified, the researcher then breeds many identical knockout mice and measures the drug response on the knockout mice versus the unaltered mice, so-called wild-type mice. So a typical result in drugs in the brain involves behavioral observations. For instance, knockout mice for the mu-opioid receptor specifically lack responses to certain types of pain, as one would expect for a receptor for morphine. Mice that have either been knocked out for the alpha-4 subunit of nicotinic receptors or the beta-2 subunit of nicotinic receptors fail to self-administer nicotine. Now, we'll talk about self-administration and its importance in animal models for addiction in a future lecture. What about the dopamine transporter? Dopamine transporter knockout mice are hyperactive, and we discussed in a previous lecture that dopamine transporter knockout mice can take up less dopamine. And sure enough, they show less response to the dopamine transporter blocker, cocaine. And they also self-administer cocaine less. We won't have time to talk about self-administration of cocaine in this course. What about cannabinoid receptors? Well, cannabinoid receptor knockouts don't differ much from normal mice. However, they lack 2 effects of externally applied THC and externally applied anandamide. First, they no longer have decreased pain responses, and they no longer have decreased heart rate in response to one of these 2 drugs. So that's a way that we can tell that the response to one of our drugs actually does go through the receptor that we hypothesize is correct. It's a question of necessity, and it doesn't enlighten us about sufficiency. Clearly, the receptor is not the only molecule sufficient for a response. On the other hand, some knockout experiments are uninformative. For instance, knockouts for NMDA receptors die at birth. So in order to understand NMDA receptors, it is necessary to make a special kind of knock-in called a conditional knock-in that lacks the NMDA receptor in only certain neuronal cell types. We won't go into that. Now, most recreational drugs act at concentrations of less than 10 micromolar, with the exception of ethanol, which, as usual, is exceptional. Caffeine acts a bit higher than that, around 10 micromolar, but a person who is legally drunk in some countries because of ethanol has a concentration significantly in excess of 1 millimolar. So ethanol's effects occur at much higher concentrations than do other drugs. Next time, we will talk about system-level effects of recreational drugs.